also from Miami that took took a lot longer. So yeah, how's it going on your end?
Good, good. Give me 1 second to my AirPods.
Yep.
Awesome. Yeah, by the way, again, I have my screen up here, so if I'm not looking down, you have to look at the screen.
Yep.
Yeah, no, it's going good. I'm supposed to take a full length 8 on Saturday, this Saturday. So I just have a couple questions. I finished all the section bank, so I guess we can okay. Sorry. Yes, I guess we can think about what to do regarding any extra like UWorld or practice questions. Um, I did okay on the section bank. I did pretty good, I think. But again, it's like I definitely did it before, so there's definitely that like aspect of doing it before. But I don't think it was that like crazy. Like me doing it before didn't influence my answer choices like that bad.
Okay.
But there is that aspect where it's like I've done it before, so
So what
there's
about
influence with that. So
the section bank volume two?
no, I'm not gonna touch. I'm thinking of not touching that until like a month out of my MCAT on March or something. But I haven't touched it.
What are you doing um in between your full lengths?
Yeah, that's what we're gonna decide, right? Because you said to first like focus on finishing second bank, which I have. So
Yeah, so I was gonna say then it would make sense to start the volume two of the section bank because what we're doing is that yeah, you're gonna do your third party full lengths um up until the end. So, what will and that'll be on the weekends. So what will we could do like during the week would be like either like UWorld like targeted content, like based on the stuff that you got wrong from the full lengths that you're taking. Um, but but uh higher priority would be finishing up the section bank volume two. So because
um,
this is still oh wait.
I'm in March. That's the thing. I'm all the way in March.
Okay, sorry. I think um got it. Okay.
Yes.
Um honestly I still think that because wait, I forgot if you had UWorld or not.
I don't have it, but I don't mind buying it though. I can buy it right now if you wanted me to. I can go right now and like buy it right now, so I have no issue with it.
Yeah.
Because
Okay.
like I feel that right now, like compared to last time, and I really want to make sure I do this time is like a lot of targeted like review. Like if I get 20 questions wrong on thermodynamics, I want to like spend all that time going through that.
Got it.
And I feel like that might be helpful. But
Got
I
it.
mean,
So yeah, so whatever questions that you get wrong from your practice tests, the in your tab, the CP content, BB content, those tabs that I added.
give me 1 second. Let me share my screen. Okay. Where? Here?
Yep.
Okay.
Uh I didn't show you this before, right?
You did, and like I tried to go through it, but I'm gonna be honest, I was like so overwhelmed going through it. I was like, whoa.
Oh, no.
So I don't really know how to.
All,
What do you think?
so all you do, so like, okay, you said question about thermodynamics or whatever.
Yeah, I was just giving thrown an example.
So just do control whatever the Mac equivalent is for control F, thermo. So and then we see that kinetic versus thermodynamic. And then we have um yeah, yeah, bio
Thermodynamics. Okay.
and then and then underneath that for the third one, you have all of this stuff. So
Okay.
you gotta like here, I can do that like let me do yeah.
Oh, thanks.
So now you see, so this is the chapter for thermodynamics, right? So this is what, so again, this is from the AMC content outline, so it's directly from the source. And this gives you the definitive uh like list of things that are fair game on the MCAT.
Yeah.
So your workflow would be like this. It'll be like you do your practice test, and let's say you got some thermodynamics questions wrong. You go to this and you look at what you need to know for thermodynamics. You need to know what the system state function is. You need to know the zeroth law, first law, PV diagram, all of this stuff, right? So that's what you need to learn. And so we can have you do UWorld passages on this topic. And as you learn that material, you can come back here and use the drop down menu to change like the thing from weak to okay, and then see how you do on your subsequent practice tests. If you're still getting thermodynamic stuff wrong, you know, you need more work on it. But if you're getting it right, then you can move using the drop down menu from okay to I think it's strong. Yeah.
Yeah. Okay.
So this way, so yeah, that'll be so does that make sense for the workflow?
So wait, let me one second. Let me log into my Blueprint account just to like look through it.
Yep.
Come on, log in. There we go. And you wouldn't suggest anything of the, I mean, I have the Q bank here, the Blueprint. You wouldn't suggest doing that, right?
Let's check it out.
Okay, wait, before we check it, I wanted to look at this. Like, I don't know. I guess we can just do this, right? Score report. So like, I guess we can look through it and then, let's say, yeah, so Gen Chem. I guess I got a couple questions wrong on stoichiometry and basic math. So I guess try to go through that section.
Okay.
Or actually, wait. Or like gases. This is what I did this time. Like I was getting a lot gone wrong on gases. So I'll just go to gases, I guess. Where is it? Gas phase. Okay. And then I'll just have to know all of this stuff. Bose law, ideal law. Okay. And then I would just do the subsequent UWorld questions that would basically test on all of these.
Yeah. So I wanted to
Okay.
do the blueprint Q bank because if that's good, then maybe you don't need to buy UWorld. So let's take a look.
I think I just opened up something randomly. Just see. Yes, but I think you go to create practice test.
So topic pool, edit topics. Yeah.
Just pick something.
Yeah, let's do like thermo or whatever.
Okay.
All right.
What do I do? 59?
Put like unlimited time.
2, 3. Oh, only 3 passages. Okay. It's weird.
Okay. Yeah, this might be something you can use. Let's try that out. Let's try it out for um like a week at least. And you know, uh uh, sorry.
What did
Uh,
you say?
let's try it. So okay, let's do this. So like, basically, you take your test, you see what questions, what areas you're, getting things wrong in. You go to the spreadsheet, you look at the content tabs, and then look up what you need to know exactly. And then you can use the blueprint QBank to make passages like this. I'm sure it can include like discretes as well, right? Uh,
Yeah, I think there was an option for that.
yeah. So then you could use that to learn to have more practice with that material. Um, and let's test it out for like a week.
Okay.
If we find that it's working, we can just have you do that. If we find that it's not working, then we can have you get UWorld.
Okay. Sounds good. Yeah, I don't know. A lot of people said I don't think I saw one good comment about it, but might as well try, see if it's somewhat decent.
Oh, what? On the Blueprint QBank?
Yeah, I don't think I heard one good comment about it, so I don't know. But
Uh,
okay. Yeah, so I guess today I'm realizing going through the BB that one of the biggest things that I'm struggling with, like content is okay, but it's the if X makes something Y happen, then how does Y affect like Z? Like a lot of that reasoning stuff. So I mean, I have a couple questions from the section bank we can go over, but I was wondering if we could also do like some like if you have a passage in mind that's like really convoluted, like a BBB passage that we could go over too.
yeah,
But that was really my plan. Okay.
that's a good thing to because yeah, I do have a good strategy for BB, like dealing with all of the because there's so many like acronyms in BB that it's very hard to keep track of it all. So let's do yeah, let's do a
Oh,
passage.
could we before we do the passage, could we go over like a couple of these questions? I think there's only like three of these section bank questions, if anything, if that's okay.
I mean, we can go over the passage and I can show you the methods, but why don't you
Okay, all right, let's okay, sure then. That's okay. We can do that then. That's fine.
yeah, but why don't you open it with the Jack Weston extension so it doesn't look horrible like this? Like it makes no sense for the AMC to like why would they do it like this, right?
The thing is, every time I do that, for some reason, it doesn't allow me to do it in Jack Weston, unfortunately. But I can try.
I'll show you. So,
Wait. Oh, start. Oh, actually, wait.
oh,
Oh,
uh,
oh my God.
just yeah,
Oops.
yeah. That's
I did not.
that's what we want. Like we want see how the format of it is
Yeah.
on the MCAT, but for some reason the AMC people who make the MCAT have it in some format where it's the answers like on the bottom, the answers like on the top right. It's like it's not two separate frames.
No, I feel you. I feel you. I didn't realize there was a start extension button, but that makes, thank, I'm
Yep.
glad I found it now. Okay.
Hmm.
So I guess I can go, I was having a tough time with this question, but I guess we can go through this passage and like look through how we can, um, do the proportionality and stuff. All right.
So, all right, why don't you start reading and I will interject when we have buzzwords and stuff.
Okay. The human intestinal tract is inhabited by a diverse assortment of microorganisms known as gut microbiota. These microbial populations are increasingly implicated in the regulation of host metabolism and the development of obesity and diabetes.
All right.
The catabolism of dietary fiber by gut microbiota results in the production of short chain fatty acids such as butyrate and acetate, which are important energy sources for the host.
What's cat
SCF
what's catabolism?
breakdown.
All right. And so, okay. So what I'm going to show you now is what I call a schematic. And so, um, so I'm going to say, okay, fiber, turns into SCFAs through the action of GM. What do you think that stands for?
Oh, the gut microbiota?
Yep.
Okay.
Okay, continue.
Such as butyrate and acetate, which are important energy sources for the host. SCFAs also act as signaling molecules with the ability to bind and activate G protein coupled receptors. Okay.
So now the SCFAs would be like a ligand that can bind to a GPCR, right?
Mhm.
So look, I'm going to turn this into a circle, and then I'm going to draw a GPCR receptor. Okay,
Mm.
right?
Okay.
Okay.
The GPCR 43, is expressed in white adipose tissue and is not expressed in muscle or liver.
All right.
Okay.
Now I'm going to specify for the GPCR that it's GPCR 43, right? See what I'm drawing?
What's up?
You see what I'm drawing?
Yeah, I'm looking at it.
Yep. And then the GPCR 43 is in what's WAT?
Okay. White adipose tissue.
Yep. Good.
Researchers generated GPCR 43 deficient mice, analyzed the contribution of GPCR 43 to energy regulation.
All right. So this is some mutant with what property?
It doesn't have that G coupled protein receptor, basically,
Good.
so it can't bind to the SCFA.
Good.
Wild type WT and GPCR 43 mice were fed a standard diet or high fat diet, and their body weight in grams was measured weekly for 16 weeks. Okay.
So what are they saying here?
So basically at this point, I think that I guess there's like four groups. So one wild type had the standard, and then there was a negative, which don't have it, and they were also fed the standard.
No, no.
There
So
was also
I mean, like
oh,
before that figure, like what are they saying here in the experiment? Like what are they doing?
so they're basically testing how these mice, the wild type versus the GPCR mice, without the GPCR mice, how their body weight fluctuates based on these diets. So I guess check for
So based on
energy regulation.
based on the diets as well as whether they're wild type or mutant, right?
Mhm.
Okay. So now figure one shows body weight of wild type and mutant mice fed a standard or high fat diet for 16 weeks. So now this is just something that I call a greater than hierarchy. So what I mean is, first of all, there's two different types of variables. So whenever we compare one variable, like we want to keep the other one constant. So what I mean is
Okay.
if we want to, so we want to look for the two things. You want to see wild type versus mutant, which one would have, let's say higher body weight. And then we also want to figure out standard diet versus high fat diet, which one has the higher body weight.
Okay.
So which one do you want to find out first?
I guess we can do a standard because it's on top.
So you want to do the diet.
Yeah, we can do the standard diet, I guess.
No,
Because
so
you said com
I mean, so we could compare wild type to mutant, or we can compare standard diet to high fat diet. Which one do you want to compare first?
Oh, okay, okay. Sure. I guess we could do standard diet versus high fat diet.
All right. So now, in order for us to compare that, we would need to be consistent with, for instance, comparing wild type standard to wild type high fat diet, right?
Okay,
And then mutant standard to mutant high fat diet. See what I mean?
okay.
Keeping one variable constant so we can compare the other.
Yeah, yeah, I see what you're saying. Okay.
I'm going to write like body weight. So you said you wanted to do the diet part first. So
Yeah, sure.
let's compare wild type standard to wild type high fat diet. This way they both are wild type, so any difference is going to be caused by the diet, right?
Yes.
Okay. So what do we observe?
I guess, oh, sorry, to zoom in. Um, yeah, so I think yeah, uh, it's a circle and triangle, so the high fat diet seems to have a higher body weight than the wild type.
Wait, then what?
The high fat diet seems to have a greater than the standard wild type,
Okay, because
right?
yeah. All right, now let's see if it's true for the mutant.
Okay, so for the mutant, the GPCR high fat diet. So, oh yeah, I guess we can cover that. Oh, sorry. The high fat diet for the GPCR 43 is greater than the GPCR 43 for the standard.
Good. So we can just leave it like this, right? High fat diet greater than standard. But if this was only true for wild type, I would write like in parentheses wild type like that, right?
Yeah.
All right, so now let's compare wild type to mutant. So that means we would
Okay.
keep the diet consistent, right?
hmm
So we would compare what to what?
You can compare the first two and then the last two.
Okay, go for it.
So in the standard diet, when the standard diet is constant, the wild type and GPCR. So in this case, it seems that the GPCR 43 without it is greater in body weight than the wild type.
Yeah, so I'm just gonna write M for mutant instead of the full GPCR 43 minus minus.
Okay.
So, okay, mutant's heavier, right? Now, that's for standard. Now, what about for high fat diet?
Yeah, so for high fat diet, it's going to be the mutant again, it seems that it's going to be higher than the mutant's going to be higher than the wild type.
Good. So we can say that the high fat diet compared to a standard diet causes weight gain, and the mutant compared to the wild type. Oh, sorry. So the first thing, I don't know if I messed up my words, but high fat diet produces a higher body weight than standard diet, and
hmm
mutant has a higher body weight compared to the wild type.
hmm
Okay, so that's our greater than hierarchy. And the thing that I wrote on that I drew on the right, I call that a schematic.
The right, yeah, with the SCFA binding to the GPCR 43.
Yeah. So now let's think a little bit, right? So the mutant means it does not have GPCR 43, right?
hmm
So that means that if you do have GPCR 43, right? um, and the SCFA binds to it, you should lose weight, right?
Yeah.
Now, let me ask you this. What if so the mutant would be getting rid of that, right?
Hmm.
Now, what if we got rid of the gut microbiota?
And this whole process wouldn't be able to happen.
So would you get what effect would that have on your weight?
I guess that would increase the weight, right?
Exactly. So that's what a schematic enables you to do. It enables you to see like if you affect things upstream and downstream of things, right?
Yeah.
Okay. Um, all right. Cool. Now continue.
Next, researchers examined the relationship between GPCR43 function and insulin sensitivity. Adipocytes from wild type and GPCR43 without GPCR43, mutant GPCR43, wild type adipose tissue were incubated in the presence and absence of both insulin and acetate, and the uptake of radioactive glucose was measured.
Okay. So now let's evaluate figure two.
Mm. I guess in the wild type, without insulin and without acetate, glucose is at this level,
Okay.
but still without acetate, I mean insulin, and they add acetate. There's really not much increase in glucose uptake.
Yeah, so anytime you have overlapping error bars, it means that there's no, statistically significant difference between the two.
Okay. Even if this little star isn't there, like let's say if this was like all the way up to 19, and we can say that it's statistically significant even without the little star.
Well, I mean, most of the time the asterisk is going to indicate to you what is statistically significant, right? But
Mm,
but yeah, like if these are statistically significant because the error bars do not overlap.
I
So
see what you're saying.
you had this, up here with the error bar there, it would be statistically significant. So they would put an asterisk there. Um,
Oh, okay.
if for some reason you see that they don't put an asterisk there and the error bars do not overlap, I guess you can say, well, two scenarios. If they had, let's say this bar is up here, so the error bars do not overlap, and let's say there's no asterisk,
Hmm.
then you can say, okay, this is statistically significant. Okay. Now, if the asterisk is present, but it's not, the asterisk isn't here, then we can say that this one on the left is not statistically significant. So the reasoning there is if there's no asterisk at all, then there's nothing that will tell us if something's statistically significant or not, except if the error bars do not overlap. But if you do have the asterisk, it means that it's not like, it means like that they didn't forget to put it. So if they're not putting it for something, then that must mean that that thing is not statistically significant. But at the end of the day, yeah, you're not going to have that. Basically, an asterisk will show statistically significance, and you won't see an asterisk if something has overlapping error bars. So,
Okay. Okay, interesting. That makes sense.
okay,
Um, so yeah, so here with the acetate, even in the mutant, it seems that it's the same.
okay,
So far I feel like acetate probably doesn't do much.
good,
But here wild type, so when they don't put acetate, insulin seems to, we have an increase in glucose. But
good.
then the second we add acetate, then there's a decrease in glucose. So, hmm.
So now, probably the most useful thing is, and we probably went over it before, but proportionalities.
Yes, we did, but yeah, I mean, you can go over it again, of course.
Yep. So I would write insulin is proportional to um, glucose uptake, right?
Okay.
Because in this bar, we add insulin and we have um, a rise in glucose uptake, right?
Yes.
Okay. Now, if we have insulin and acetate, seems like the amount of glucose uptake decreases, right?
Hmm.
So how can we express that?
I guess insulin is like inversely proportional to acetate, or I
To
guess glucose uptake is inversely proportional to acetate, maybe?
So that's where it can get a little tricky
Okay.
because if that, so you're saying acetate is, bless you. So you're saying acetate inversely proportional to glucose uptake
Yeah, so I'm guessing as acetate increases, glucose uptake decreases.
as acetate glucose uptake increase what?
No, if acetate is added or acetate levels are increased, then glucose uptake is decreased.
Okay. So an inverse proportion you're saying.
Yes.
So yeah, so that's the tricky thing. Because if that was true, why for these two, when we have acetate, do we not see a decrease in glucose uptake?
Mm-hmm.
Right?
Mm-hmm.
So we can't say that acetate is inversely proportional to glucose uptake. What we can say is acetate is inversely proportional to insulin sensitivity, right?
Mm,
Because
okay.
if glucose, if glucose raises the, sorry, if insulin raises the uptake of glucose,
Okay.
the acetate doesn't directly reduce the uptake of glucose. The acetate reduces the sensitivity of the insulin, making it so that insulin cannot cause that much of an increase in glucose uptake,
Right.
right? So
Hmm.
it's a subtle kind of thing here, but yeah, we want to be careful there. Um, like let's see if I can find, I'm going to think of like an analogy. So, um, all right. So insulin, same thing, glucose uptake, right? Now, if I used an inhibitor of insulin, that would cause a decrease in glucose uptake, right?
Inhibitor. Yeah, because glucose can't bind to insulin. Okay.
Now, if now does that mean that the inhibitor itself reduces the amount of glucose?
Not quite.
Because if you did an experiment where you didn't have any insulin, but you just had the inhibitor, right? The glucose levels shouldn't change, right?
Hmm.
But if you have insulin and the inhibitor, then they should change, right? So
Yeah.
the key thing here is that an inhibitor can inhibit the enzyme that causes glucose uptake, meaning you get a smaller amount of glucose uptake, but it doesn't directly reduce the amount of glucose uptake. It just
Okay.
inhibits the thing that causes glucose uptake,
It's the thing that causes it. Yeah.
right? So
Mm.
good. Okay. Now, what do we see for the next bar?
Yeah. So it seems that with the mutant, with insulin, it's already pretty high.
I mean, and that
Actually, well, I mean, compared to like the other mutant here without insulin, it seems to increase it to some degree, the glucose uptake.
well,
Um,
so same thing with wild type, right?
oh, right, right.
So insulin proportional to glucose uptake would be true for both wild type and mutant, right?
Okay.
All right.
And even with the acetate, it seems that it doesn't go down.
Uh-huh.
So
So
there seems to, in a GPCR 43, I guess there's no really like insulin. It doesn't really have that effect on insulin sensitivity for some reason.
so now in parentheses, I'm just going to write wild type. That means acetate is inversely proportional to insulin sensitivity only in the wild type.
Okay.
So these are the three main strategies. The schematic here that lets you visualize the pathway, which can then help you analyze things upstream and downstream of the pathway. On the left here, we have just a regular greater than hierarchy where we determine that, what type of diet causes an increase in body weight, a mutant compared to wild type, which one would have a greater body weight. And then the most common one that you'll probably use would be the proportionalities, which I wrote here on the right. So those are the things that we will take down as, in our notes as we analyze a BB passage because BB just has a lot of acronyms. Like this passage isn't even that bad, but a lot of the passages, bless you, a lot of the passages have just tons and tons of acronyms and like this inhibits this, this activates this. It's impossible to keep track of all of that. That's why writing out a proportionality kind of reduces it down to like kind of like just a logic truth false kind of thing, right?
Okay. Yeah.
Maybe I could even write GPCR 43, meaning having it, not the mutant. Um, or actually I could just write wild type as well, but this would be inversely proportional to body weight, right?
Yeah. Because
Um,
it would bind and cause the reaction to, I mean, the whole cascade of events to happen.
yep, I could even say gut microbiota is also inversely proportional to body weight, right?
Mm.
Yep, right? Makes sense?
Yeah.
All right, cool.
Hmm.
Now, if you want to take a screenshot, you could take a screenshot. Okay. So, all right. What is this question asking us here?
Which table shows the expected body weight of wild type and GPCR negative negative, or mutant, mice fed a high-fat diet while housed for 16 weeks in a conventional condition or a germ-free condition, where the gut does not become colonized. So I guess they want to compare how body weights would differ between these two conditions, between if they're conventional or if they are germ-free.
Okay, let's start with the wild type. What would it? Okay, so what are your thoughts on this?
So the main thing I guess we gotta look, I guess to consider is the one condition where the gut microbiota are there, and then I guess another condition where the gut microbiota are not there.
All right, so go on.
I guess we can compare both of these in the conventional and then both of these in the germ-free. Um,
Let, let's
so in
let's
this, oh, sorry.
let's start with just yeah, wild type in these conditions.
Okay. So wild type in conventional, um, I feel like I feel like comparatively, the germ-free would probably have more body weight, right? Because if this wouldn't be happening. And we also wrote that they're inversely proportional to body weight.
All right. So for wild type conventional and wild type germ free, you're saying what about the body weights?
The germ-free is going to be probably higher.
Good. So we want to see wild type germ free body weight greater than wild type conventional body weight, right?
Hmm.
All right. Now mutant. Tell me about that.
So if they're mutant in the conventional sense, um, they probably increase because they can't bind to this. So if that's the case, then the body weight would probably be pretty high. And if there's no germ-free, then it's just going to be, I'm thinking it's either going to be the same or it's going to be higher.
Mm-hmm.
But I'm feeling it's probably going to be a little bit higher too because this isn't being broken down anyway, so it's going to be even more. It's like another on top of that.
So what can cause a decrease in body weight?
Binding to the GPCR43 or the gut microbiota.
So so binding to the SCFA is binding to GPCR43, right?
Yeah,
So if you don't have any GPCR43, how much binding do you have?
nothing,
Now, if you don't have SCFAs and you don't have GPCR43, how much binding do you have?
nothing,
So
more nothing.
more nothing,
Or actually, probably nothing, right? Yeah, I'm going with nothing, right?
nothing is zero, right?
Yeah, can't go more than zero.
I mean, there's negatives and stuff.
Yeah, but I guess not here.
Okay, what are we then expecting to see in our answer?
So I guess the GPCR43 conventional and germ-free are going to be probably around the same body weight.
Right?
Um, and then for the wild type and GPCR43, no, sorry, the wild type in the conventional and germ-free, the germ-free is probably going to be a little bit higher
Excellent.
than the conventional.
Excellent.
Oh, let's see. We can go through this. So I guess we can say, yeah, the germ-free is going to be a little bit more than the conventional. This would make sense because they're like very close to each other, so I don't know about that. Um, here,
So there,
yeah, this makes, oh, sorry.
these are close together, which you don't like, right?
Yeah.
And then what about these?
Oh, um, yeah, you want the GPC of 43 to be pretty close as well.
And this shows a decrease, right?
Yeah, that wouldn't make any sense.
So is this a good choice?
Probably not.
Okay.
And then here we said that, yeah, the germ-free is a little bit more, but then here it's conventional and germ-free are the same.
Okay.
So that kind of goes along with what we said.
All right.
Here, yeah, I'm not sure about this because they're similar. You'd want the germ-free to be again a little bit more.
Okay.
I'm breaking it down. And then here you are on the similar terms here.
Okay.
But again, that's not what we're, this kind of negates it, so no. Uh, and then here is kind of the opposite of what we said, right? We want the germ-free to be a little bit more.
Okay.
Conventional and germ-free are a little similar here, but
Are
that
they?
negates it. I feel like they're not far off, right? I mean, although it says decrease, but I don't think they're far off because even here they kind of decrease, right? A little bit.
So, I mean, this goes change. This is different by two. This is different by two. This is different by one. This is different by six. So I
Okay,
think
this is
this is a decrease. Yeah.
okay.
So for C, what if conventional was 39 and germ-free was 41?
Still no, right? Because it's still just a very
Mhm.
small. It's not too much to say that. It's not too much to say that it's like yeah. I don't think it's still enough.
Good. All right.
Okay.
So what's our answer?
B right. I think yeah,
Yep.
B.
Yep.
Can't change it, but
Yeah. So
yeah, I can.
so these types of passages could be difficult in that we not only have to interpret the data for in the passage, but even in the questions, we have to interpret data, right?
Yeah.
And that's hard to do unless you have a systematic way of doing it. And the systematic way that we did it was, well, we drew the schematic, we drew the or wrote out the greater than hierarchy, and we wrote out the proportionalities. So that way, it's kind of like the equivalent of like a car's map.
Yeah.
I mean, it's different, but it both serve the purpose of passage, taking a passage and distilling it down to the important stuff um, that helps us understand it easier. Um, because, you learn in psych soc, like our brain's working memory can hold 7 plus or minus 2 bits of information at a time. So it's just literally impossible to have all this, analyze all this stuff in your head. So writing stuff out is always what you want to be doing. And that enabled us to come up with an answer so that when we looked through the choices, we knew what we were looking for because we thought about it and came up with an answer. Um, Instead of just going right into the choices.
Mm.
Because I guess you did that and you got D. Um, what was your reasoning for that?
I don't remember. I think my reasoning was, I think it was just the confusion with the body weight. I think I really, yeah, I got stuck with just interpreting this stuff, I think. That just made it a little hard to go back and see that relationships. But
Got it.
yeah, I tend to,
Because
yeah, sometimes I don't really make it a point to draw a lot of these schematics and stuff.
yep, yep.
So
Yeah, so it seems like you chose the one that has a decrease in body weight for germ-free for
yeah,
both wild type and mutant. So, but yeah, that's like the paradoxical nature of like the MCAT. Like, there's a time crunch. Well, you didn't do this time, so shouldn't be that. But in general, there's a time crunch, but if you rush, you're gonna get things wrong. So we want that, middle ground kind of thing. So, all right, good job.
okay. This one was, oh, 30. I was gonna use the bathroom real quick, but I'm gonna pull it up this question.
You don't have any other questions for that?
Yeah,
That?
no, I did. I was gonna pull it up and I'm gonna use the bathroom real quick, but
Oh no. So I was gonna. So you don't have any more questions from the passage we just read?
no, no. I don't think there's any more. I think I understood this.
Oh, okay. All right. Wait,
Yeah,
that one's wrong.
I'm sure. Okay. Uh,
You can use the bathroom.
okay. Compared to untreated wild-type mice, antibiotic treatment of wild-type mice is most likely to result in like a decreased body mass. Okay, so if you use antibiotic, wild-type mice are essentially able, they have this schematic, so they'd be able to break it down into CFA, bind to it. So
What does it mean, antibiotic treatment of mice?
yes, it'll probably destroy and kill all the gut microbiota. So there wouldn't be enough to break down the dietary fiber into the SPFAs.
Yep.
So first sense choice is increased plasma butyrate levels, uh, probably decrease levels because yeah, so you're not breaking it down into SPFA, which is one of these. So yeah, that wouldn't be yeah, probably decreased. Probably not decrease body mass because yeah, you're not getting any of this, and it's not able to bind to the GPCR43. So probably increase body mass, increase volume of adipocytes. Um, yeah, that kind of falls in the line of it. And then The decreased insulin sensitivity in adipocytes. Seems a far-fetched. I mean, let me see. Decreased insulin sensitivity. So decreased insulin sensitivity, acetate. I think it would be increased insulin sensitivity, right? I think so. It decreased.
What decreased insulin sensitivity? Like,
And oh yeah, so an increase in acetate would probably decrease the insulin sensitivity, right?
okay.
But if you don't have antibiotics, you're not breaking it down to FCFAs, so you wouldn't be having any of the acetates forming.
Uh-huh.
So you probably have decreased acetate.
Yep.
So you probably have you should the answer choice would probably say increased insulin sensitivity, right?
Good.
Okay. So I guess it would be C, right?
Yep.
Okay.
And I just want to make sure for the other ones. So let's just look at 8 and
Sure.
so 8. Um,
Least likely to be found on the transmembrane domains.
yeah.
Yeah, so typically transmembrane are just going to be a lot of nonpolar
Yep.
amino acids. So yeah, so aspartate is the only one.
All right. Next one. Um,
Compared to wild type mice, which experimental group of mice is most likely to remain lean? Oh, you wanted to just check or you wanted to read through it? I'm sorry.
oh no no. So it makes sense why this answer is C over here.
Okay, yeah, it's been a while since I looked, but I mean, I can look at it again. Let me see.
Mhm.
Yes, I mean, the reaction would be, if it's overexpressed, this whole reaction would be happening more and more.
So oh, so
What the? Sorry.
yeah.
Okay.
So antibiotics would kill the gut microbiota, which means you're not going to get the SCFAs, which means you're not going to get anything binding to the GPCR43, which means that you're not going to get you're going to gain weight, right?
Hmm.
And mice treated with a GPCR43 antagonist means that you're making the GPCR43 messed up, which means higher body weight. And then D is an example of something downstream of the GPCR43. Just like getting rid of the gut microbiota is upstream of the GPCR43, the GPCR43, when the SCFA binds to it, produces a second messenger cascade inside of the cell, right?
Yeah.
So if you inhibit the second messengers, you are doing a downstream thing, uh, effectively making, the equivalent of as if the GPCR43 wasn't working or wasn't there, right?
Hmm.
Good. And this one makes sense.
Yeah.
And does this one make sense?
Yeah, muscle wouldn't change because all we know is that all of this happens in the
Yep.
I remember it was like the adipose. So and then this one, yeah, acetate. It's basically just a downstream target. Yeah, it would probably be doing the same thing,
Yep. Perfect.
similar to this. Okay.
Great job.
And I think I had a question on 30. This one was weird. Um, kind of understood it, but it was yeah, it's like certain like probability stuff like this. Yeah, this I guess it's something we can go over. Oh yeah, so in a species of beetle red flower, red body color is dominant to brown. Two red beetles are crossed and produce 31 red and 9 brown. Two red F1 are crossed. What is the probability that both red and brown will appear in F2?
All right, let's see. So I hate these, by the way.
Yo, same, 100%.
Okay.
Worst questions.
Red body color is dominant to brown. So if you're heterozygous, you would be what?
Big, I guess we can go with big R. Yeah, big R and small r.
Oh no, I mean, what color? If you were heterozygous, what color would you be?
Red.
Okay, okay. All right. So two beetles are crossed and produce 31 red and 9 brown offspring, and that's the F1 generation. So what can you tell me about that?
So when I did this, I kind of understood that if the two red wheels are crossed, they're three, so 31 red and 9 brown. So it's probably, I realized, okay, that's like a 3-to-4 ratio. So it's probably, both of them are probably going to be heterozygous.
Good,
The F1 generation is probably going to be heterozygous.
good. So we can say for the parent generation, like R, um, oh, well, at least it isn't a dihybrid cross.
Yeah.
I hate those in particular. But yeah, okay. So that lets us be able to tell that it's we're crossing two heterozygous ones. Um and for the F1 generation, we would have a big R, two big R little R's, and one little R, which is the brown one, right?
Yes.
All right, now if two red, oh, okay, this is brown and these are red, right?
Hmm.
So now if they cross two red F1 beetles, what is the probability that both red and brown beetles appear in the F2 generation, right?
Hmm.
So, so that means okay, we are not going to use this, right? Because it's not red.
Yeah.
We can use these, right? Right?
Yeah.
Now, if they had like let's say they crossed a big R with heterozygous, you would get big R, big R, big R, big R, big R, little r, big R, little r. Is this likely um
No, because
what?
you get all red.
Good. So we're not so we can do the two heterozygous again. Big R, big R, big R, little r, big R, little r, little r, little r, right?
Hmm.
So that can give you some that can give you both red and brown beetles in the F2 generation, right?
Yeah.
All right. So this is also, the cross for the parents. So if we're trying to figure out the probability that both red and brown beetles will appear in the F2 generation, um, first in the F1 generation, right, we would get wait. Oh, okay. So F1 generation gives us, big R, two big R, little R's, and then one little R, right? And they
Hmm.
are crossing two red F1 beetles, which are just these. So that's 3 out of the 4, right?
3 out of the 4, yeah.
3 out of the wait. Um,
2 out of 4, right? Oh no, wait.
sorry. It is 3 out of the 4 of the offspring, but when we're crossing them and they must be red, um, and we also need the F2 generation to have both red and brown. This would have to be this heterozygous cross again, right?
Yeah,
So, since we have this much in our F1 generation, and we're only choosing red beetles, we have a total
beetles.
of 3 out of 4 red beetles, right? But in order to have both red and brown beetles in the F2 generation, we can't have the big R big R. So, so out of the 3 red beetles, only the heterozygous ones, when mated, can have both red and brown offspring, right?
Yeah.
So this is a 2 out of 3 um proba probability. Um, or rather, so to get this is a 2 out of 3 probability because you have 3 red beetles and you can only choose the heterozygous ones, so that's 2 out of the 3 that you can uh choose.
Okay.
And same thing with this one, right?
Mm.
2 out of the 3. And in the F2 generation, we would get that. We would get both red and brown beetles if we did this cross. So when you have um when you have probabilities of, like, I forgot if it was like let me see one sec. So if the question is what is the probability of, like, let's say, 2 events happening, how about this? The probability of getting heads on a coin toss is 1/2.
Hmm.
The probability of getting heads on 2 coin tosses would be 1/2 times 1/2. You multiply it. If we're trying to figure out the probability of an event and another event, you multiply the probabilities. Whereas,
Said one and another. Okay.
yeah, if it's and, you multiply the probabilities. If it's or, you add the probabilities.
Okay. What would be the case in that for the heads and tails if it's or?
Oh yeah, so the or could be like, let's say you get heads uh one time and then, or well, I guess, so let's say it's heads or tails. That
Okay.
would be 1/2 probability for the heads plus 1/2 probability for the tails, which will just be 1 or 100% probability that you'll get either heads or tails. But if you got, let's say, heads and heads, it would be a 1/4 probability.
Mm, that makes sense.
Because if you add probabilities, you get a larger number, but if you multiply probabilities, aka fractions, you get a smaller number. And it's always less likely for additional events to happen compared to fewer events.
Yeah, it'd be pretty hard to get heads 10 times in a row. So,
Exactly, right?
okay.
And that would just be 1/2 times 1/2 10 times, or 1/2 to the 10th power.
Hmm.
So,
Yeah, I think this question was confusing me because it was like, I think I wrote something.
oh yeah.
Yeah, I
So,
don't get why the question is like asking for the fact that it's asking for the parent's genotype, that really confused me. So I was like, I mean, it's gonna be just I just read the probability that both red and brown beetles, so I figured it would just be like 3/4 ratio. But
well,
like the fact that it said parents, I was just so like, I don't know.
where do you see parents?
Or I mean, I guess the F is two red F1, right? That's why I figured that's the parent, right? Or maybe I'm mixing up my terminology.
That's the parent right here.
Mm,
So okay, they're telling us that they took two red for the parents. They took two red beetles and crossed them. So that means that the parent genotypes could either be big R, or big R, little r, right?
yes.
So if they were both big R, you'll get all red, right? If you cross big R with big R, little r, which is the first thing that I did here, you also get all red. But if you do big big R, little r, you get red, 3/4 of it red, and 1/4 brown, right?
Hmm.
So if they're telling us that the F1 generation is red and brown, the parents must be heterozygous, right?
Yeah.
Okay. And then they said if you take two red F1 beetles and you cross them, what's the probability that you'll get both red and brown beetles in the F2 generation? Well, the probability of, so first of all, from this, right, we get what I wrote here. You get one big R, you get two big R, little r, and you get one little r. Now, the one little r is brown, so we don't care about that at all because we're crossing two red F1 beetles, right?
Two red F1 beetles. Yep.
And if we used big R, we wouldn't get any of the brown beetles that they're asking for. So we must cross the heterozygous F1 beetles, right?
Hmm.
So they're asking what's the probability, like if you cross these, what's the probability that in the F2 generation you'll get both red and brown beetles, right? So so we have three total red beetles, but only two out of the three red beetles would produce offspring that are both red and brown, right?
Yeah.
So for the, let's say, the father, the probability of that being like heterozygous here is 2/3 because we have three total red beetles and only two out of those red beetles can produce offspring that'll be both red and brown, right?
Hmm.
And same thing for this one. And so we're crossing them, so we multiply uh, you know, the probabilities because they're asking, you know, both red and brown beetles. So we multiply the probabilities to get 4 out of 9.
Okay. Yeah, that makes sense.
Mhm. So,
76. Okay.
so for so there's always going to be these types of problems. So I think the easiest way to kind of go about them is, well, no, I guess that if you have and, you multiply the probabilities. If you have or, you add the probabilities, and yeah, and that's pretty much that. Yeah.
Okay. Let's just think, how would it change if it's a dihybrid cross? Would that do anything more?
Oh, sorry, say that again.
If theoretically this was some sort of dihybrid cross, how would that? And I guess we can try to think about that.
Oh yeah. So that's going to, I mean, there's ways of doing it. And then there's also like the phenotypic ratio of 9:3:3:1. Uh, but that would make things quite a bit more complicated because you'll have ultimately, so dihybrid cross would be like 4 by 4 like alleles.
Yeah.
It'll give you 16 total possibilities, and you'll go from there essentially.
Wow. I guess I gotta pray that they don't do that.
Yeah, that's very vague answer that I gave, but we'll probably see one.
Yeah, this one was weird. I didn't call this one. I still didn't understand after I read the answer choice.
Um, so okay, let's see. Um, I'm just gonna read the passage quickly. So resistant. This is the only question you have for this passage, right?
Yeah, everything else was on this was fine.
Okay.
I think. Yeah.
Resistant
Jesus.
hypertension is a type of hypertension that remains uncontrolled, despite treatment with antihypertensive medicines. It is hypothesized that the signaling of pro-renin, an inactive precursor of the renin component of the renin-angiotensin system in the central nervous system, plays a crucial role in the pathophysiology of this vascular disorder. Researchers have shown that the binding of prorenin to its receptor PRR in nerve cells of the hypothalamus results in a conformational change in prorenin and activation of its catalytic functions. Okay, so prorenin binds to PRR and activates, right? Are you there? Oh, I think you froze. Okay. All right. So let's see. Okay, so when Pro-Renin binds to PRR, um, it gets activated. C17XA, a substrate analog, which binds to the active site of activated Pro-Renin, has been used to inhibit catalytic activity of Pro-Renin. Um, what type of inhibitor do you think C17XA is?
Probably competitive, right? If it binds to the active site.
All right, to investigate the roles of prorenin and its hypothalamic receptor and pathways leading to resistant hypertension, researchers cloned the gene encoding PRR, wild type PRR, and constructed several PRR variants. So again, this is something that you will see over and over again. Passages that talk about an enzyme, about amino acids at the active site of the enzyme, and to figure out which amino acids at the active site are important. They're gonna make mutant variants in which they replace an amino acid with some at a specific residue with something else, and then they look at how that affects the kinetics of it. So the dissociation constants KD of prorenin for each of the PRR variants were determined table 1. So we have wild type KD 0.034, variant V109E, pretty much the same KD. W140L, we seem to have like 5 times the amount of KD. D201N, we have like um more than 10 times the amount of KD. And then A269K, we have, oh, a really tiny KD, right?
Hmm.
All right, let's, all right, mouse neuro A2A cells were transfected with either cloned wild type PRR cDNA or an empty vector as controls for 72 hours. Cells were then treated with PBS buffer control, an angiotensin 2 receptor antagonist, losartan, prorenin, or a combination of losartan and prorenin for 30 minutes. The levels of reactive oxygen species, ROS, what's that, by the way?
Uh, Reactive oxygen species. I think they're created from the breakdown of hydrogen peroxide, right? I think so.
So there, you know what free radicals are?
Yeah, yeah. Free radicals are. I mean, aren't they? They're made from hydrogen peroxide, right?
I mean, that could be an example, but tell me what it
Oh.
exactly.
Um, in the context of the mitochondria, that seems to be somewhere I see it.
So, okay,
That's about it. That's really about it.
so you know how atoms have lone pairs
Yeah.
because electrons like to be in pairs, right?
Hmm.
When an electron is alone and it's not in a pair, it's like, like what the word incel means?
Yeah.
It's basically like that. Because, like, okay, so like without going too much on a tangent, like most like school shooters and stuff like that are these types of people. Because yeah, like that would make people really angry. So a free radical is an electron that's alone and it's super pissed off and it's very reactive and damaging. So yeah. So that's what a free radical is. And you've probably heard about antioxidants.
Yeah, I think there's a vitamin that does that. I think it's vitamin K. I think I could be wrong. Something like that, right? I
So
think. I don't know.
an antioxidant is something that gives free radicals an electron so it can stop being so destructive.
Okay.
So and yeah, reactive oxygen species are essentially free radicals. So all right. So they measured that, and which is an intermediary molecule responsible for hypertension. Um, so okay, figure one, relative levels of ROS in neuro 2A cells 3 days after transfection with either an empty vector, black bars, or wild type PRRC DNA, gray bars, treated with various compounds. Okay, y-axis levels of ROS, which we take to be a proxy for hypertension, right?
Mm-hmm.
So in the control, the empty vector, the wild type PRR has a lot more ROS. And when you use losartan, which is an angiotensin 2 receptor antagonist, doesn't seem to change the levels of ROS, right?
Yeah.
When we have Pro-Renin, we see an increase in both empty vector and wild type PRR, right?
Yeah.
Right? And then if you have Pro-Renin plus losartan, it seems like it brings the wild type PRR amount down, but it doesn't really change the empty vector part, right?
No.
Okay. And oh, this could be an example of, when they don't give you asterisks,
Mm.
then you can see, if the error bars overlap or not, and they don't seem to be overlapping anywhere here. So this all should be statistically significant.
So it's okay to make that. It's like again, it's okay to make that assumption.
Correct.
Okay. Okay.
All right, now let's see. So, oh wait, based on the data presented in figure one, overexpression of PRR, let's see, increases blood pressure. Okay, so ROS proportional to hypertension, aka proportional to blood pressure, right?
Mm-hmm.
And for everything, even control, the wild type PRR um has a higher amount of ROS, which means a higher blood pressure, right?
Yep.
Now, well, I can get rid of Roman numeral 3, right? Because that says a decrease in blood pressure.
Yeah. There's no increase. There's no decrease anywhere.
Good. So, all right, and now if it's angiotensin 2 dependent or independent, well, if it goes up in the control, right? We're not that would be an angiotensin 2 independent pathway, right? And if we use losartan is a antagonist of angiotensin 2. So losartan would make the angio. Oh wait, I think it's the other way around then. So losartan inhibits angiotensin 2, right?
Losartan inhibits. Yes.
So if we see when you have losartan that we have higher blood pressure, That would be this through an angiotensin 2 independent pathway because losartan will block the angiotensin 2 pathway, yet it still causes an increase in ROS, which means an increase in blood pressure, right?
I mean, okay, so, but like, it's not that much though, right? How could we say that it's independent, right?
Oh, well, what are you asking?
If both of these bars are the same,
Yeah.
wouldn't it be still angiotensin dependent?
Well,
Because it doesn't really change much?
well, that's the thing, right? Because we're not really comparing like what we're comparing is wild type PRR to the empty vector, right?
Okay.
Empty vector means it doesn't have. So see, it says that these cells were transfected with either wild type PRR or an empty vector. The empty vector does not have PRR, right?
Hmm. I mean, even if they don't have empty vector, the cells still have some level of PR, right?
Uh, shouldn't they shouldn't have that?
Hmm. Okay.
Like what? Okay, like what? Tell me what you're thinking.
My thinking is, if I understand it a bit more now, because like you're saying that both of these ROS,
So the empty vector does it make sense that the empty vector should not have any PRR? Oh, you think you froze again.
I'm really sorry about that, Shane. I don't know what's going on, man.
You're good. You're good.
Okay, I just connected to my phone, so it's like on a hotspot. So I'm just gonna stop my video. I'm sorry,
Yep.
but I'll share my screen now. Hopefully, it continues to work.
Sure. So, um, so okay. They're telling us here that these cells either were cloned with or added they added either the gene for PRR or an empty vector. So that means that the empty vector does not have PRR and the gray one it has PRR, right?
Okay.
Now, in the control, we see that there's higher levels of ROS, which means higher levels of hypertension or blood pressure, right? Simply by having more PRR, right?
Okay.
Because the black bar has no PRR and the gray bar has
Hmm.
PRR, right?
Okay. Sounds good.
All right. Now, next, when we introduce losartan is an antagonist for the angiotensin 2 receptor, which means that if you use losartan, your angiotensin 2 stops working, right?
I was working. Okay.
Now, we see from figure 1 that when you use losartan and therefore you stop the angiotensin 2 from working, you still have high increased blood pressure, right?
I'm so sorry. Could you say that last part again?
Hmm. So so if you use losartan, you don't have angiotensin 2 working anymore, yet we still see an increase in blood pressure, right?
Okay.
Right? So that would mean that there's a pathway that is independent of angiotensin 2.
And you're comparing the fact that increased to the control, right? Or you're not comparing it to like the empty vector, sorry.
I'm comparing it to the empty vector because the question stem says overexpression of PRR.
Okay, now I see what you're saying. Okay.
Now, this is not the wild type PRR is not overexpressing it, but it's expressing it, and the empty vector is not expressing it. So
Okay.
if there's an increase in ROS when you have PRR compared to when you don't have PRR, I can reasonably say if we overexpress PRR, that you'll get even probably more ROS, right?
Mm, yeah.
So the fact that the fact that ROS levels increase when you block angiotensin 2 means that you can increase your blood pressure through an angiotensin 2 independent pathway, right?
Okay.
So, um, does that make sense though? Like,
Yeah, now I think I kept looking at the control, but I see what you're saying. Like still, even even with losartan, and that we're inhibiting or really stopping the receptor, like there's still compared to the empty vector where there's nothing, right? There's still more reactive species. So,
yep,
and we said that that relates to blood pressure, so it's like it's still high. So I see what you're saying.
yep. So that would be the angiotensin 2 independent pathway. Now, let's say in the control in which you have um angiotensin working, right?
Mhm.
So angiotensin is working in the control, and we still see that increase in blood pressure. So that could be
Mhm.
this, right?
Mhm. Okay, I see what you're saying. That makes sense. And then what about this column? This column doesn't seem make sense to me. So with Pro-Renin,
Mhm.
it's like does losartan, yeah,
Yeah.
so why is the level of reactive species with Pro-Renin and losartan decrease here?
Yeah. So the Pro-Renin seems to increase blood pressure for both the wild type and the empty vector, right? So we can't
Mm-hmm.
so there's no way for us to be able to tell if Pro-Renin, doing what it's doing here, is dependent or independent of angiotensin 2, right?
Mm-hmm.
So yeah, so that doesn't help us out there. Um, and then in the combination, when Pro-Renin and losartan, it seems like the amount compared to just Pro-Renin, the amount for the wild type PRR decreases. Um, and then compared to the Pro-Renin, the empty vector amount stays the same. But compared to Losartan, right, we have kind of like the same amount for the wild type PRR. So the Pro-Renin doesn't tell us anything about and whether it's angiotensin 2 dependent or independent.
But why does I mean, I guess without that in, like, I see that part, but just looking at the graph, like, why is it that adding losartan decreases it, the reactive species?
Uh, well, we could say that um let's see. Well, let's see. Okay. So considering that Losartan and control are the same, it seems like Losartan is not something that causes uh an increase in ROS, whereas Pro-Renin we can see causes an increase in ROS. So the combination of the two could be, you know, the Pro-Renin will cause it to increase and the Losartan will cause it to decrease. So you have it in the middle kind of. Right? It's almost like this is like insulin, acetate, and then, or rather, acetate, acetate, insulin, insulin,
Mm,
acetate. Like
I
the
can't see
ac
the connection.
yeah, the acetate or Losartan in this case alone does not cause a decrease in ROS. And like insulin, Pro-Renin causes an increase in ROS. So if you have a combination of the two, Pro-Renin and Losartan, it's like insulin and acetate, which means that it lowers it the ROS levels compared to if you just had Pro-Renin. So
Mm,
Losartan we can't say is inversely proportional to ROS, but we can say that it's probably inversely proportional to like Pro-Renin sensitivity or something.
okay. Okay, this makes a lot more sense. Yeah, like how you put the insulin and acetate, that makes it a lot
Hmm.
easier to understand that.
Yep.
Oh, all right. Let's see. I think that's really it.
Uh,
Oh,
anything else from this?
yeah. What's up?
Oh, I was gonna ask anything else for this passage?
Let me see.
Look at 70.
Not really. Based on which amino acid residues are involved in binding to the protein. 2, 3, and 4. 140, 201, and 269. Yeah, because even with this, it still doesn't really change the KD, but these all change it, so they probably are important.
Good. Let's look at the next.
Exhibit C inhibition. Yeah, because it says it's active site. So without the inhibitor, it's only K. The Vmax should be the same, but Km should increase.
Good.
This we just went over. Based on the data in table 1, which amino acid residue with prorenin most likely interacts with the residue? 201. See, I don't think it's talking about the mutant, but if it's aspartic acid, it's probably going to be something that's polar. Then we have arginine.
So so the aspartic acid is charged, and if something is polar, it has a partial charge. Think about it like that.
Polar is a partial charge. What do you mean by partial charge?
So carbonyl group is a good example,
Mm-hmm.
right? There's a dipole here with that's facing this way towards the oxygen, which means the oxygen has a partial negative and the carbonyl carbon has a partial positive. So something that's polar won't be charged, but it'll have partial charges. Whereas things that are full-fledged charged are like they'll exhibit the same properties that a polar molecule will exhibit, but even more so,
Okay.
right? So, yeah.
Sorry.
So position 201 is what's that?
Aspartic acid.
All right. And what can you tell me about that? Is that,
It just has a carboxyl group. Um, Depending on the pH, it will be deprotonated and protonated.
I mean, tell me about like
PK, I guess.
regular physiological pH.
Oh, yeah, it should be. It probably should be the deprotonated.
So it'll have a charge.
Mm-hmm.
What charge?
Just a negative charge.
Okay. And what would likely, I don't mean the choices here, but what type of thing would uh interact with it?
Anything with a positive charge?
Good. So so you said arginine because it was polar, but it's arginine because it's positively charged, right?
Yeah, I see what you're saying.
Like glutamine is polar,
Mm-hmm.
right?
Yeah.
So so that's the thing. And that's usually a gripe that I have about when people memorize the amino acids, like if they memorize the polar ones versus non-polar, like you should be able to tell if something is polar by looking at the molecule, but also by recognizing that it's the same thing as being charged, except it's partial charge. Okay.
Partial charge.
Par
Okay.
Anything that's polar will have to have a will have to have partial charges, which means that it'll have to involve like a highly electronegative atom for connected to something that has a lower electronegativity.
Anything polar needs to have a partial charge. Okay.
Yeah,
I mean, well, glutamine doesn't have a charge, right? It's polar but doesn't have a charge.
it does. Glutamate. So
Because glutamine just has the amide group, right? And but it doesn't have
what's this?
a carboxylic acid.
And when it loses its proton,
Carboxylate, I think. Yeah, carboxylate.
carboxylate, right?
Mm-hmm.
So what is this?
Aspartic acid.
And when it loses its proton,
Oh yeah, it's as I think it's aspartate, right? Aspartate.
so glutamate
Mm.
just glutamic acid when it loses its proton.
No, I meant to say, but glutamine, right? No, glutamine doesn't have a charge, right?
Oh, okay. I thought you were asking about glutamate. Correct.
No.
Glutamine.
Okay.
So you can kind of think of it like this. Glutamate, aka glutamic acid, you know,
Mm.
its side chain will have a carboxylic acid group. If you replace that with an amide, you will get glutamine. And
Yeah.
Aspartic acid, if you replace the carboxylic acid side chain with an amide, you'll get asparagine,
Mhm.
right?
Okay.
Yeah.
Okay. That was a good point that you brought up with the ick and the AIDS. That makes more a lot of sense because I guess they're already considering it as the in the charged form here.
Yep.
Okay.
Yeah. Be able to be, understand that they're, that they're the same thing essentially. They could also say like, uh the things can also be like uh like let's say it was like something like that. Like
Hmm.
this could be called a carboxylate salt,
Hmm.
which means that it just, it's bound to like a sodium, which means that it readily, like in water, it'll readily turn into the carboxylate ion, and the sodium will just be a spectator. But that's what it would be called if it's a salt. But yeah.
Okay. Cool, cool. And then the last one. Yeah, I might have got this wrong, but I understood why because it was like cloning of it. I kind of forgot how to do it. But yeah, because cloning, you start with an mRNA and then use reverse transcriptase to make a complementary DNA strand. But I didn't realize that the complementary DNA strand will also need a getting its other strand as well. So yeah, I probably would need DNA ligase to like bind it all together,
Uh, well,
right?
so okay, when I read the cDNA thing, I was actually thinking about asking you because that's kind of like a buzzword. Um,
Mhm.
but tell me about cDNA.
Yeah, so complementary DNA is typically made from you have an mRNA molecule, which has already basically gone through its modifications and stuff.
Okay.
And then you have reverse transcriptase that comes in and it can make a essentially, yeah, complementary like DNA strand essentially from it.
Okay.
Um, and then I don't know what exactly, but that mRNA strand can get degraded,
Yeah.
and then that complementary DNA strand creates its own other, uh, yeah, another strand, I guess, to become double stranded
So, and that other strand is made of what?
DNA.
Okay. So, what do you need?
Probably DNA polymerase,
Yeah.
right?
Um,
Mhm.
you need the reverse transcriptase. What about the ligase?
Yeah, you probably need the ligase to bind the phosphodiester bonds, right?
Uh-huh.
So yeah, so it makes sense. You probably wouldn't need the or you wouldn't need a RNA polymerase because you're not trying to make RNA.
Correct.
So that makes sense. Okay. Yeah, I think that was it. I wish we could have gone through a BB passage, but unfortunately I have to go. But I mean, hopefully maybe next time we can try to go through a
Oh, we went over like a
Hmm?
did
Oh yeah, I did. I mean, we went over these two, but I also wanted to go on like even more practice for a second because I really want to try to get better at this. But no, but I think today was great though. Like I feel like I understood, and I'm gonna try to. I'm definitely going to try to do really be more aware and try to make those schematics and those charts and stuff. So
yep.
yeah,
Your schematics will help for visualizing pathways. Um, and the greater than thing, self-explanatory, and the proportionality thing is going to help you the most with dealing with acronyms. And yeah, you just want to go slow and systematic, and yeah, you'll improve that way. So you're taking a test on Saturday?
yeah. On something full length blueprint 8. That's the plan.
Got it. Okay. And then I'm seeing you next week. Yeah, on Monday, it seems, on Tuesday, Wednesday. Yeah.
Wait, what?
Oh,
Tuesday and Wednesday?
I see you for Monday the 16th, Tuesday the 17th, and Wednesday the 18th.
Uh, okay, I think I might have missed a mistake. Could you take out the Tuesday? Just Monday and Wednesday would be okay.
Uh-huh. Okay. All right.